Okay, so today we're going to talk about gene expression and how we regulate that expression. Because remember that all the cells in our body have that same genetic information, that same DNA, but they have different roles, and that means we're not using all of the DNA all at the same time. So how do we regulate when do we have and in which cells do we have certain regions of the DNA being used to express protein, and when do we have it not being used? So this is all at gene expression and regulation. Okay, so there's a couple of different places we can regulate this gene expression. So remember, we get the information from the DNA, we transcribe it into RNA, and then it's translated into a protein. So we want to regulate what proteins are made depending on the situation. And so we can regulate that at the transcription level as well as the translation level. So first we're going to talk about how we can regulate it at the transcription level. Okay, so again, gene expression, this is where we're using that information from the DNA to transcribe and translate it into a functional protein. That's DNA to RNA to protein. So different types of chemical reactions are going to regulate gene expression. One way we can do this is by using regulatory sequences. These are stretches of DNA that are used to promote or inhibit protein synthesis. You might have remembered from that PhET simulation that we did earlier in the unit where there was a regulatory sequence of DNA before the actual coding region of the DNA that gets turned into the protein. This is what we're talking about here, where that extra bit of DNA at the beginning regulates when we are not using that DNA. So here we can see we have our introns and exons, and then we have some regulatory sequences right before it. And they're going to impact how the DNA is transcribed. We have regulatory proteins that are also going to be involved in this. So those are proteins that bind to different regulatory sequences. Again, we can have ones that inhibit, and they kind of block that RNA polymerase from binding and making the RNA product. Or we can have proteins that that help this process. So together, they're all going to control transcription. We can also get at what we call epigenetics. So epigenetics is the reversible modifications of DNA or histones. Epigenetics is a more recent field that we've been looking at more. This is where we can have changes to the DNA that we can change over time. So it's not actually changing the sequence of the DNA, it's adding things to the DNA, or sometimes it's affecting the histones. Remember, the histones are those proteins that the DNA is wrapped around when we have it condensed. Okay, these can also affect gene expression. So DNA that is tightly packed together is going to be inaccessible. So you can imagine about DNA that's way in here. It will be difficult for RNA polymerase to reach it so that we can have transcription happening. We can also have DNA modifications. This is where we add different methyl groups or things. It's just adding little chemical flags kind of to the DNA, and that's going to signal transcription or inhibit transcription, depending on the case. So methylation is an example of this. Acetylation, all these are examples of adding a methyl group or an acetyl group. This is just a chemical group that's added onto the DNA. Development can also be regulated. So development of an organism is coordinated by changes in gene expression. This is the idea of cell differentiation. So as an organism grows and develops, we don't need all of those cells to be exactly the same. We need them to differentiate into different roles. And this happens through our gene expression and the regulation of gene expression. So here is showing the very early stages of a cell before it develops into an organism. And we eventually get to where we have different types of cells. You don't have to get into all the details of this diagram, but just knowing that we have different types of cells that are going to go on to do different functions. And this is all regulated through gene expression. And this is, internal cues, but also external cues are involved in this signaling as well. So this is accomplished by the expression of cell type specific proteins. Yes, so it's different depending on the cell type. It will express different proteins. So we can think of these also kind of as switches.